Mary, for the nice introduction, and thanks Applied Stem Cells for this opportunity. And to everyone who is listening, thank you all for your interest and taking the time out of your day to participate in today's webinar. In the next 30 minutes or so, I'll be talking to you about how to use autobioluminescent cells to reduce the cost and complexity of your optical imaging applications. Before we get started, I'd like to give you an outline of the topics that will be covered today. I will start with a brief introduction to autobioluminescence, explaining what it is, where it comes from, and its adaptation for function in human cells. Then I will move on to the comparison of the novel autobioluminescent technology with the traditional bioluminescent imaging technology that you're probably already familiar with. I will be discussing the similarities and differences between the two approaches and also the pros and cons of each technology. And once we have the basic background information squared away, I will present some examples of applying autobioluminescence in vitro cell-based assays as well as in vivo small animal imaging. And I'll be discussing how it can reduce the cost and complexity of these applications. At the end, I'll briefly touch base on one unique application of autobioluminescent imaging, which is its expression in stem cells. The first question many people ask is, what do you mean by autobioluminescence? And my go-to answer to that question is, autobioluminescence is the autonomous production of light without the need of optical excitation, which is required for fluorescent imaging, or the need of adding exogenous substrate, which is often necessary for traditional bioluminescent imaging. Autobioluminescence is found natively in many marine bacterial species, a classic example being the bobtail squid symbiote Alibir fisheri. However, for system development purpose, the autobioluminescent genetic pathway from the terrestrial bacterium Photorhabdus luminescens is the one that was chosen for expression in human cells. This is because with an optimal growth temperature at 30 degrees Celsius, as opposed to the 20 degrees Celsius for most of the marine species, the autobioluminescent signal from Photorhabdus luminescens is stable at human physiological temperature 37 degrees Celsius. And there is currently known toxicity of Photorhabdus luminescens to human cells. The genetic pathway for autobioluminescence, which I will refer as the Lux cassette, is consisted of multiple genes that encode proteins to produce both the luciferase enzyme and the luciferin substrate. The Lux A and Lux B genes encode a luciferase dimer that function just the same as a traditional luciferase. So in the presence of molecular oxygen and reduced flavin mononucleotide, FMH2, the Lux AB luciferase converts the aldehyde luciferin substrate to fatty acid and produce a light signal at 490 nanometer during the process. Slightly redshifted compared to nanoluc and ranella luciferase, the 490 nanometer wavelengths allows it to be multiplexed with these reporters as well as traditional firefly luciferase or other fluorescent reporters. The most distinctive feature of autobioluminescence is its ability to produce its own luciferin substrate within the cells. This is made possible due to the expression of the remaining genes in this pathway. So the Lux C, Lux D, and Lux E proteins work together to convert endogenous fatty acids to aldehyde, therefore providing a continuous supply of the otherwise non-scavengable substrate for the luciferase. So all 5 genes working together, as long as the Lux pathway is properly expressed, light can be continuously produced, so you don't need to worry about signal decay over time as you would in other bioluminescent systems. Despite it being a complex multi-gene cassette, the autobioluminescent pathway has been re-engineered by linking the LUX genes with with multiple viral 2A linkers to enable all genes to be expressed as a single construct using a single promoter in human cells. So now you have the complexity and coordinated expression of a full pathway with the convenience and ease of use of a single gene. From a molecular biology standpoint, this means that there's no need to change your cloning, transfection, or selection protocols, and you can empower your cells without developing new methods. So now we've covered the basics of autobioluminescence. People, especially those of you who currently use bioluminescent imaging in your research, often are curious about how this new technology compares with traditional bioluminescent imaging technology. So let's take a quick look at the basic pros and cons of each approach. Traditional bioluminescent imaging has several advantages, such as ultra-low background in cells and animal tissues. There's a variety of multiple luciferase platforms that you can choose from, and there's a pretty good signal intensity, so you can detect a very small amount of target cells. However, because it requires exogenous substrate for light production, there are concerns of experimental variations due to variable dose kinetics, substrate uptake rate, and the biodistribution of the substrate within the animal. For in vitro cell assays, using traditional bioluminescent imaging often requires sample destruction, so you would have to prepare multiple samples for different time points of your interest, and then that limits your flexibility to collect data. Autobioluminescent imaging has the same ultra-low background advantage as traditional bioluminescent imaging, but it can do so without any investigator interaction because the cells can supply its own luciferase substrate. The light signal is generated autonomously from the cells without sample destruction, which allows you to continuously track the signal dynamics over time from the same sample. And by getting rid of the step of adding exogenous substrate, the autobioluminescent imaging significantly reduced the hands-on time, so with an increased ease of use. And of course, autobioluminescent imaging has its own disadvantages. It's a relatively new novel technology, so it hasn't been used very commonly in the literature. It is also a larger genetic construct compared to other single gene bioluminescent reporters, so there might be some difficulties in terms of molecular cloning. And because of the cells self-generate their own substrate instead of being saturated with exogenously added substrate, the signal output is relatively lower. Another very common concern of the autobioluminescent technology is whether or not the constitutively active expression of the full pathway places a metabolic burden on the cells. So it has been shown that that's actually not the case. Despite the continuous production of light, cells expressing the pathway do not display altered metabolic activity levels relatively to their wild-type host cells. So as you can see in this figure, this was done by an ATP content assay looking at autobioluminescent cells and wild-type cells at right after plating in 24 hours, after 24 hours of growth, and there's no significant effect on metabolic activity was observed. And this was been done in multiple tissue types. So we looked at the actually how much it would save us for using autobioluminescence. So this numbers shown up here could change based on your institution and how much your technician is being paid. But in general, we, based on our calculation, I've seen maybe a 30 to 40% savings in cost because of getting rid of substrate and reduced hands-on time from your technician. So for example, for in vivo imaging, you just need to inject your cells and anesthetize the animal and start imaging whenever you want. But if you're using traditional bioluminescent imaging that you would have to inject a substrate every time you have to take image, that would significantly increase the hands-on time as well as the cost for the substrate. Similar for in vitro cell-based assays, because the cells can be imaged continuously over time, you don't have to prepare different cell samples for each time point. So that's actually a lot of saving on hands-on time for sample preparation as well, in addition to the savings for the substrate. So here's just a table summarizing the difference between autobioluminescence and bioluminescent imaging in terms of cost. I just want to highlight a few things on this table. So for animal imaging, because autobioluminescence imaging does not require you to inject another substrate prior to each imaging time point, so there's no repetitive needle sticks, which means that your eye cook would be really happy because that's increased animal welfare. And also by reducing the injection, you're actually reducing a lot of error, increasing your statistical power for experiment. You could actually use less animal to get a better result. So now we've covered the basics. I would like to spend the next time on explaining how autobioluminescence can be used for a lot of applications. So I'll start with in vitro applications. The typical, very classic application for autobioluminescent imaging is to track metabolic activity. This is because in order for the cells to produce a light signal, the Lux pathway has to be successfully transcribed and translated. The cells needs to be metabolically active to provide all the cofactors for this reaction. And also because it uses SMH2, it is very sensitive to the metabolic activity of the cells. So the light output correlates strongly with metabolic activity, and this was consistent with other metabolic tracking assays such as the traditional MTT, MTS assays. But you can do so by monitoring the same cell population over time instead of preparing separate samples for each time point. Again, shown on the left graph, it's just a typical graph of using autobioluminescent HEK293 cells to track cytotoxic compounds. So you can see that because the cells can be tracked continuously over time, it gives you the power to look at dose effect as well as temporal effect just in one plate in one assay. You don't have to worry about preparing different samples for each time point. And the one graph shown on the right is a very neat example that I have that I'd like to share with you guys is that, so this figure shows here is use autobioluminescence to track pathogenic bacterial infection. So by labeling the human cells with autobioluminescence, and you can infect the cells with different dose of pathogenic bacteria, you can continuously track how the metabolic activity of the human cells change in response to infection by this bacteria. So imagine you would have to do this with traditional bioluminescent assay. Not only you would have to prepare different samples for each time point, which correlates to each point on that graph, but also you would have to worry about isolate the human cells from the mixed population, which could be a very laborious and time-consuming and probably not very efficient as well. And because of the Lux cassette, the autobioluminescent gene cassette is a genetic system, so the genes are being passed along to daughter cells during cell division. So if you put the genes under a constitutive control, it's a good tool to track cell population growth, so track the change of cell numbers. So this graph shown on the left here is actually using estrogen responsive autobioluminescent T47D human breast cancer cells to track the cellular growth in response to estrogenic compound exposure. So same idea again, because you can keep the cells intact, you can track the population over time and repeatedly on every day. So So you get more data with actually less cellular preparation of work. And because even though it's a full pathway, it's been engineered to function as a single gene. So you can pop out the constitutive promoter and put in a promoter of your interest. You can actually be looking at the activation of your specific promoter of interest very easily and continuously looking at the time. So as the example shown on the slides here is by replacing the expression of the autobioluminescent pathway under a TET-ON promoter and to look at doxycycline detection. And you can pretty much replace it with whatever promoter of your interest, and then you can look at the same way continuously. So the reduction in cost and complexity is really obvious for a large-scale library screen. When you have thousands of compounds to test, you would want to do an ultra-high throughput 1536 well plate, but you don't really know what time point is the best time point to look at. So with traditional bioluminescent assay, you would have to prepare multiple 1536 well plates to look at different time points. But if you use autobioluminescent cells, you can look at the cells continuously or repetitively at any time points of your interest. So if you look at the graph here, graph A, B, and C actually are produced from one simple plate. It just look at this plate at different time points, and if you just use one plate for a traditional ATP content-based assay, you would only have one time point data. So without increasing preparation work, you actually get more data if you're using autobioluminescent cells. And most importantly, the assay metrics such as signal to background ratios, the coefficient of variations, and Z factors remain very consistent over time, and also it's very relative to traditional assays. And you get similar results as well. You get it correlates really well with traditional assays that you're already familiar with. Another application for autobioluminescent imaging is in 3D cell cultures. So an obstacle for 3D culture imaging is, if you're using traditional bioluminescent imaging, is you have to worry about the uptake rate or the distribution of the substrate to the core of your structure, because the cells on the exterior surface and the inner core have different exposure to the substrate. But you wouldn't have to worry about that for autobioluminescent imaging, because each cell, regardless of its location within the structure, has the same ability to produce a light signal. You also wouldn't have to worry about excitation signal if you're using fluorescent imaging, because you would have to worry about the cells within the inner core of the structure doesn't get enough excitation compared to the cells on the exterior surface. And again, because the cells can stay intact and metabolic active over time, you get this ability to track the metabolic response of the cells to different scaffold materials or configurations over time very easily. So after talking about the in vitro applications, I'd like to give a few examples about the application of autobioluminescent imaging for in vivo studies. So here's a very brief comparison of how the autobioluminescent imaging compare with traditional luciferase systems. So on the top of the graph, you'll see that you can inject the autobioluminescent cells into animal, or you can develop transgenic animal models already expressing the autobioluminescent phenotype, and then you're good to go. You just put your animals in your equipment and hit start and get an image, and then if you don't like the image, just take another picture. Or you want to take a picture every day, no problem at all. Just place your animal back into the instrument and run it again. If you're using traditional substrate-dependent bioluminescent cell lines, such as those expressing firefly luciferase, you would do the same injection, cell injection, but you don't get a signal yet. You would have to inject another luciferin, and to trigger the light reaction, And because of the injection, you would have to worry about, okay, what's the best route for injection? Do I do IP? Do I do IV? Or, do I do sub-Q? Or what's the dose that I have to use? Maybe I have to do a pre-experiment to optimize the dose for the specific animal. Or you would have to worry about what if the luciferin substrate interact with my compound of interest and give me false results. So not only that, and after this one image, if you don't like it, you have to, wait for the previously injected substrate to clear out and then re-inject your substrate and then image again. And if you want to do it a week later, you have to repeat the process again. So, just as I said, the advantage of using autobioluminescent imaging is that you only require a single cellular injection at the very beginning, and then you can image your animal subject at any time point throughout the entire experiment period. And because there's no substrate injection involved, the signal is not affected by substrate quality or dose efficiency, because those become irrelevant. Don't have to worry about substrate uptake and clearance rate because they're also irrelevant. And you don't even have to change your equipment because it's using the same equipment. Your existing equipment is good at looking at autobioluminescent signal as well. And it can also be multiplexed with other reporter systems. If you can have a different cell type expressing a fluorescent label, then you can look at them simultaneously. And typical example for in vivo imaging is for oncogenic applications. So you can use the bioluminescent signal, the light signal, to look at the tumor burden. So here's a graph comparing autobioluminescent tumor cells and traditional bioluminescent tumor models. You'll see on the autobioluminescent tumor cells, actually the signal correlates better with the tumor volume than the traditional luciferase assays. And this is because if you think about the tumor, it's a 3D structure. So same thing with this 3D cell culture, the cells inside the tumor won't get as much as substrate as the cells on the exterior surface of the tumor. So that's why there's a large variation between the bioluminescent signal and the volumes. And because of the autobioluminescent cells get rid of the need to substrate and get rid of all the concerns about substrate uptake distribution, you would have a tighter correlation between your signal output and your tumor volume. Same thing as we said earlier, because the cells are constantly expressing this pathway, will it affect the tumor growth? So, and the answer to that is probably not, because it has been shown that the autobioluminescent tumor models are the same as bioluminescent tumor using luciferin requiring reporter system in terms of weight and growth rate. And for animal applications, if you want to look at tissue-specific promoter activity, or if you want to look at your synthetic promoter of interest, and because of the pathway it's engineered as a single gene, you could just easily swap out a promoter and look at whatever promoter activity you're interested very easily. So at the end, I'd like to briefly talk about a unique application of autobioluminescent imaging, which is expression in stem cells. So there has been evidence showing that expression of the autobioluminescent pathway in stem cells does not induce differentiation, which is good, so your stem cells maintain as stem cells. And also it does not affect the capability to differentiate. So autobioluminescent stem cells can also differentiate into whatever lineage is supposed to be, just as the wild type stem cells. And because of that, the autobioluminescent technology allows you to track the process of stem cell differentiation, and you can look at the basal metabolic activity of different lineages at the end very easily. again showing that the autobioluminescent signal is maintained throughout the differentiation process, but you can see the different metabolic activity rate in different cell types after differentiation. For in vivo tracking, same idea here. If you want to look at the efficiency of your stem cell therapy, you would inject the autobioluminescent stem cells into your animal and look at the trafficking of the stem cells. Very easily you can look at if it's homing to the site of interest, or how do they survive, do they die, do they differentiate into other cell types. Or if you want to look at, for tissue engineering purposes, you could preload the autobioluminescent stem cells on a 3D scaffold and implant the 3D scaffold in your animal and looking at cell retention, and you can look at cellular health at any area of interest. So with all that, I just want to give you some quick rundown of conclusions of today. So the take-home message for today is that autobioluminescent cells allow you to perform the exact same experiments that you're already doing and using the exact same equipment that you already have, but with reduced cost and complexity of your work, because now you can repeatedly assay your samples at any desired time point without any extra preparation work. The autobioluminescent expression system, because it's been packaged as a single gene, function as a single gene, it is very easy to use. It can be introduced into both immortalized or stem cell lines, or introduced into animal models, so you can use them to improve the efficiency of any assay of interest. The system also allows users with no previous imaging experience to quickly pick up the technology and very cost-effectively to begin using luminescence in your lab for a variety of applications.
Thank you, Dr. Xu. Now we will take some questions. The first question is, how stable is the autobioluminescent signal following multiple passages?
We have found that the autobioluminescent signal remains pretty stable over time, over multiple passages, always long enough for us to collect all the data that we need. However, because it is pretty much impossible to account for all cellular changes that could result from normal cellular growth and cellular maintenance, I would be confident saying that you can maintain your active culture for up to 10 to 15 passages. At this level, you can ensure that the autobioluminescent signal remains stable, but can also minimize the chance that any intracellular changes could affect your assay results.
Let's take another one. Does the system work in formalin fixed tissue section?
Unfortunately not, because in order to produce autobioluminescent, the cells needs to be in a metabolically active state, because the cells need to transcribe and translate the genes to produce the proteins, and the proteins need to be functional, using all the endogenous metabolites to produce life. So right now the system would not be functional in fixed tissue sections.
Thank you, Dr. Xu. One of the attendees is asking, is there any special equipment required to assay these cells?
No, you can use your existing equipment that you already have. And this is because the autobioluminescent signal is simply a light with a wavelength of 490 nanometers. So if you have a plate reader that can read bioluminescence, you can probably use that to read autobioluminescent signal. And if you have a CCD-based camera for animal work that can also read bioluminescence, you can use it to read autobioluminescent as well. I don't think you even need any other modifications to do so. And if you have a dedicated fluorescent detection equipment, if it can be, operated without an excitation source and with an open emission filter to collect all the light coming out of it, it will likely be compatible with the autobioluminescent signal as well without too much modification.
Okay. The next question is, have you transfected any primary cells? And does it require lentiviral packaged vector?
So yes, the short answer to that question is we have successfully transfected primary cells with the autobioluminescent pathway, gene cassette. We've done so by using a variety of methods such as traditional chemical transfection, electroporation, as well as lentiviral gene transfer technologies. So in order to transfect those difficult to transfect primary cells, I would highly recommend using a helper insertion technology such as applied stem cells target technology to increase the efficiency of chromosomal integration.
Thank you. Here's another one. How well does the autobioluminescent signal correlate with alternative assays?
Its short answer is pretty well. I actually kind of touched it a little bit in the slides. The continuously generated signal from the autobioluminescent cells allows them to be used in place of many other common or more time-consuming and more expensive assay formats. We've observed very strong correlations between autobioluminescent signal dynamics and other cytotoxicity assays such as MTT, MTS assay, the ATP content-based assays. Similarly, because for estrogenic compound detection, this assay provides a very good alternative to the destructive E-screen assays, as we observed very similar EC50 values. However, because the autobioluminescent technology is relatively new, I would recommend that you start by performing a side-by-side comparison between your assay of interest and the autobioluminescent assay, just to check how well they correlate each other. And if nothing else, this will at least make you appreciate how much less work it is to use autobioluminescent technology.
Okay. Thank you again for attending our webinar. Don't worry if we didn't get to your questions today. We will be sure to follow up with you via email. If you have any follow-up questions about our products or services, please send them to info@appliedstemcell.com and we will connect you with one of our experienced technical specialists. Applied Stem Cell already has ready-to-go products in human colon, kidney, and breast cancer cell lines expressing the autobioluminescent signal. However, we also specialize in generating customized autobioluminescent cell lines of your choice. Please contact us by phone or via email if you have any specific need for autobioluminescent cell lines. We also have the autobioluminescent vectors for you to purchase, so you can generate your own autobioluminescent lines in-house. Within the next few days, we will send you a copy of